Okay, so final exam review. I did get an email with some questions, so I figured we'd start with those and then we'd go from there. All right, so first question was, will the tissues chapter be on the final? And no, it will not. It is just the 4 systems. So it's everything after the tissues chapter. Hopefully that clears that up. Okay, so the second question is how spongy bone formation occurs before the medullary cavity formation in the endochondral process. So, I mean, I can explain it. Let me pull it up and see if that helps. But I mean, the thing is, like, we didn't really cut. Give me 1 second. Okay, endochondral ossification, formation of spongy bone before medullary cavity formation. Let me pull it up. Okay, so first I just want to point out, like, this is the level at which you need to understand it, okay? So, you know, you guys don't really have to know the why of it, but I understand how going through it helps, you know, like that totally makes sense for me. So I think that the easiest way to understand it is to appreciate the fact that when we first have bone formation, what's happening is the same thing that happens with the bone lengthening process. So, you know, we have cartilage formation. The cartilage calcifies and dies, right? That's like part of this process. So it's doing its thing, it expands, it calcifies, it dies off because it's not supposed to have a calcified extracellular matrix. The blood vessels come in and the osteoclasts come in and they start dismantling the dead cartilage, and the osteoblasts use the dead cartilage to build bone tissue kind of on top of it as it's getting replaced. So we're using this dead cartilage framework to construct the bone. That doesn't necessarily give us cortical bone. That's going to give us more of a spongy bone if we're on the inside. And then as we get expansion in either direction, we can start to actually form the medullary cavity, right? But we have to, like, essentially clear out the spongy bone that formed first. The cortical bone is going to form as that bone collar. Not quite the same thing as the bony callus of fracture repair, but similar in that we're making it on the outside. So the cortical bone's forming from there. So they're kind of doing this at the same time, by slightly different parts of the same process. Okay, but I mean, the thing is that it seems to be easier to make structures and then take some of them back than to start with something like a cavity, if that makes sense. But like really, you don't need to know the details beyond this. We don't have time to cover it. And so I know it's weird, but just kind of keep in mind that all the bone that's internal to the compact bone outside is going to be spongy bone, if that helps. All right, let's see. Next question was, does bone form straight from connective tissue in intramembranous ossification? Okay, so bone. Okay, so here's the thing with a question like that. The answer is yes, but also the answer is, because remember that bone itself is a connective tissue, and cartilage is a connective tissue. So I think the question is bone forming straight from mesenchyme? Because that's what's happening in intramembranous ossification, is that we have this mesenchyme, which is the embryonic connective tissue that then becomes all of the adult ones that we've learned about. So yes, in that case, there's a direct transformation. So basically, differentiation is the term we use for when cells become more specialized. So the mesenchymal cells, which are the stem cells of connective tissue, most of them become fibroblasts because that's our most common connective tissue cell type. They're going to become osteoblasts. So they will specialize further into osteoblasts, who then immediately start secreting the osteoid, the organic part of the bone extracellular matrix. Okay, so that's the big thing with the intramembranous, is that we're doing this direct transformation, which is really cool, but it's also limiting because without the cartilage framework, all we can make is that flat bone, right? All we can do is make this little flat sheet of starting with spongy bone, and then underneath the periosteum, we end up forming the two layers of compact bone that it's sandwiching the spongy bone. Okay, so one of the reasons why like vocabulary is so important is that if you're not using the correct term, you might not actually be communicating the right concepts, right? So that's why that kind of thing is kind of critical, not only for what I try to teach you guys, but also how I ask you questions, and also how I expect you to be able to answer the questions. Is if you're giving me a term like connective tissue, it better really mean connective tissue. If you're trying to refer to a specific one, then you need to specify, because otherwise, there's a lot of them. Okay. All right, so next question is the role of vitamin D. Okay, so the question is the relationship between vitamin D and calcium in mineral homeostasis. So really though, what that question is, now I'm gonna have to find which that was like the next lecture. Oh no, it's right here, cool. So the relationship between the two of them is that vitamin D is one of the hormones that helps us maintain calcium levels in the blood. So when we look at calcium homeostasis, there's three hormones of note. There's parathyroid hormone, there's calcitonin, and there's calcitriol. And calcitriol is the hormone name for vitamin D, okay? Parathyroid hormone is the most important because it's the one that has that has the relationship where it's released directly in response to blood levels. So if blood levels of calcium decrease, parathyroid hormone gets secreted from the parathyroid glands. One of the things that parathyroid hormone does is it activates vitamin D. So it tells the kidneys, because they're the ones that do the final activation stage, like, hey, we need more active vitamin D. So the kidneys release or activate vitamin D. And since we call the hormone form of it calcitriol, now we have calcitriol circulating. And as a hormone, it does a couple of different things. So it backs up the functions of parathyroid hormone by increasing resorption from the skeleton, and increasing reabsorption by the kidneys, okay? Those are the two other things that parathyroid hormone does. It's the only one that can increase absorption of calcium from the diet. And as we kind of touched on, you know, like last week or whenever it was, we were talking about digestion and absorption. It's going to do that by increasing the mechanisms by which we're uptaking the calcium. So it literally increases movement of calcium from the lumen of the small intestine through the enterocytes. So although parathyroid hormone is far more critical, vitamin D is still super important, because parathyroid hormone can increase circulating levels of calcium by releasing it from the bone and prevent us from losing it. But the only way to actually get more calcium total into the body is by having vitamin D help us do that. Okay, so next question is about synovial joints. So let me go to that. All right, so the first question is about menisci. Oh, I made it one single section, that's nice. So let's go down to synovial joints. All right. So when it comes to synovial joints, there's your basic synovial joint anatomy, and then there's a couple of extra things that some synovial joints have. And one of the major bonus features of synovial joints is fibrocartilage pads. So fibrocartilage pads, their major function is to increase fit. So sometimes the two bones that are coming together in a joint don't actually have very compatible shapes. And so adding fibrocartilage pads is a great way to get that shape compatibility. And then as a tough cartilage, fibrocartilage also is good for shock absorption and stuff like that. So the menisci are specifically the fibrocartilage pads within the knees. And the reason why they have that name as opposed to being called an articular disc, which is what we call it everywhere else that it exists, is because instead of being a circle, it's like a crescent shape. So they just named it after its shape, sort of. So it's a fibrocartilage pad. Additionally, we do have other synovial structures that aren't joints. So the bursae and the tendon sheaths, they are synovial structures because they have synovial fluid inside of a cavity lined with a synovial membrane. But they're not joints because they are not joining two bones, okay? So when you're looking at the synovial joint stuff, this is the anatomy that we're worrying about. This is standard that we find in every single synovial joint. This is the extra stuff that we can sometimes see. So sometimes we can see these fibrocartilage pads, and sometimes we see these other synovial structures, they're always going to be nearby joints because typically it's the movement of the joint itself that creates the need for them, but they're not considered to be part of the joints themselves because they're not directly connected to them. Okay, so when you're looking at that kind of stuff, make sure you're distinguishing what category things stick into or fit into. Okay, next joint question. Do we need to know the locations of each type of joint? If you're asking me if you need to know where the different categories of joints are located, the answer to that is only if they're very specific. So if you're asking about, like, you know, sutures are only located between the bones of the skull, then yes. If you're asking, like, do we need to know which joints in the body count as like saddle joints or plain joints or something, for the most part, no. The only exceptions to that are the ones where there are very few. So like plain joints, you should know that the carpal bones and the tarsal bones all form plain joints, because that's like, as far as you're concerned, that's it. That's what we have. And we only have the two ball and socket joints. So if I feel like that's fair that you should know that the shoulder and hip joints are ball and socket. But I don't really expect you to know the other ones, you know, when they're fairly common and throughout the body. All right, synovial joint stability. Okay, let's go back to that. So I didn't spend a lot of time on it because there's only so much stuff I can talk about. And sometimes I do go into this more, but really, when we're talking about joint stability for synovial joints, there's a balance between range of motion and stability. So the things that typically increase the stability of synovial joints, such as the fit of the bone surfaces at the joint, so like how the bones come together, how tight the dense connective tissues surrounding the joint are, so mostly the ligaments holding it together, as well as the joint capsule itself. And then to a lesser extent, what the muscles and their connecting tendons do across the joints all contribute to stability. So a very stable joint tends to have less range of motion and less movement associated with it, because everything is so tightly joined together that even though it's a diarthrotic joint, it's still not like super movable. And we see that in some places, and more with certain types of joints too. So when we look at like those plain joints, that's a great example of a very stable joint with a very limited range of motion, because all they can do is glide. And one of the reasons is because, of course, the surfaces are flat. And so you can't really do anything other than glide across. But the other reason is that it's between a bunch of these little short bones. And so it's not like it's just one surface where they could do a bunch of gliding. It's these teeny tiny little surfaces, and they're all connected together by ligaments. And so all of them have just this tiniest amount of movement. But it's also really hard to like move your wrist out of alignment because you have a bunch of bones tightly bound to each other. When we look at the other side of it, the other extreme to this is your shoulder. Your shoulder has the largest range of motion of any joint in your body. In part, that's because it's a ball and socket joint, so it's got that multi-axial movement, right? It can do pretty much everything. But it's also because it has a fairly loose joint capsule. There aren't nearly as many ligaments surrounding it as in other joints. In fact, a lot of the stability of the joint itself is provided by what we call the rotator cuff, which is actually 3 tendons of muscles going across the joint. And as a lot of you probably know, it's really easy to injure your rotator cuff. And that's quite painful, I understand. And when you look at the hip by comparison, even though it's the exact same type of joint, right? They're both ball and socket joints. The hip has a much more limited range of motion because there are some very strong ligaments joining the femur across to the coxal bone, and there's a lot of muscle surrounding it as well. And so the whole joint itself is much more stable, and therefore has a more limited range of motion to it, even though the joint itself is essentially the same as the one in the shoulder. Okay, so when we talk about stability of synovial joints, we're looking at the things that make it fit together and how that contrasts with range of motion. I think that was okay, yeah, now we gotta go to other stuff. Okay, so second, or not second, but next question is about levers. Let me go to that one. Okay, so hopefully the basic idea of a lever is pretty straightforward to everybody, that we have a couple of components and that basically we're, using our bones as these rigid things, and then our joints are the fulcrums that they rotate around, and our muscles are applying force, right? And that's how we end up with movement. So the way that levers are classified is based on the relationship between the fulcrum, the force, and the effort. And so what that means is, well, here, let me just do it this way. Okay, so when we take a lever, when we take our little guy right here, and we put a fulcrum in here, okay? So there are only so many ways that we can arrange the three components of a lever system. So I can put the force, that's a little muscle there, the force on one side, and the effort on the other side, right? That's easy enough. Force, fulcrum, load. And then I can put them both on the same side. So I can put the force closer to the fulcrum, and I don't know what I'm doing, and the load farther away. Or I can put the load closer and the force farther away. Although you can run these the other way around, I can draw this and be like, oh, look, it's on this side instead, it still ends up being only three orders, right? So, right, yeah, we're going to call that effort. Okay, so you can have the effort, the fulcrum, and the load. You can also write this out like this. It doesn't really matter, but this tends to be the one that actually gets used. Means the same thing. You can have the fulcrum, the effort, and the load, and you could write it like that. Or you can have the effort, the load, and the fulcrum. Okay, so because there's only three possible arrangements as far as looking at their relationship to each other, that's how we get these three different classes of of levers. And the physics of the thing tells us that there are certain things we know just by knowing the class, which is why we care. So when we look at a second class lever, we're always putting force in our effort farther away from the fulcrum than the effort. And the consequence of that is that we're getting an increase in power for our output compared to what we're putting in, right? So it's easier to move things with second class levers because we get more force out than we're putting in. And that's, you know, why wheelbarrows are made the way they are. It's why like shovels work so well compared to, you know, having a longer handle on your shovel compared to having a shorter handle, and stuff like that. So we're getting more power out of a second class lever, always, because literally the thing that defines a power lever is that the effort is farther from the fulcrum than the load. And the same thing defines a second class lever, okay? With a third class lever, it's the opposite. So with a third class lever, our effort is always closer to the fulcrum than our load. And so we know that when that happens, we're at a power disadvantage, right? It's a mechanical disadvantage, but we call them speed levers. So third class levers are always speed levers because when your effort is farther away from your fulcrum than your effort is, you lose power, but you gain speed once you overcome the effort, okay? First class levers can go either way because when you have your lever arms separated like that, it depends on the length of each arm, right? I don't know just by having a first class lever whether it's going to be a power lever or a speed lever because I need to actually measure the distance between the fulcrum and the effort versus the fulcrum and the load. So these guys can go either way, and that's handy because, you know, this is how stuff like scissors work. And so if you really need to put a lot of force into what you're cutting, you can put your load closer to your fulcrum to get more power. And when you just need to cut something really fast, you can put your load farther away to get more speed, right? So the reason why we classify the levers is because then we know what type of lever we have. And just because, you know, in physics, just like in biology, we like to classify everything. So hopefully that helps with that. Okay, on to the next question. All right, on to muscle. Okay. Is excitability the same as conductivity in muscle characteristics? Let's see what I said before. Because they shouldn't quite be the same thing. Okay, so I think I'm going to need a little bit more information to answer this question because I'm not clear as to when we ever had the word conductivity used here. So excitability Button. So excitability is the term we use for cells that can respond to stimuli. So excitability is actually one of the characteristics that muscle tissue and nervous tissue share, because they're both really good at responding to a stimulus. Conductivity is not a term we use to describe muscle tissues. We use it for nervous tissue to describe the ability of like neurons to transmit electrical impulses. Technically, muscle tissue can do that to a certain extent. We certainly see that in cardiac muscle. Because they are technically all electrically synapsed to each other. But I'm not sure how else to answer that question without more information. Okay, so the next one is about the motor nerve varicosities in smooth muscle. But the last question I got was reviewing the steps of the contraction cycle. So let me do that one first, and then I'll do the smooth muscle. All right, so the contraction cycle is pretty cool because, like, you know, like it literally just goes around and around and around and we get muscle contraction. There is a beginning to the cycle, even though it is cycling around continuously. So the reason why we have a beginning is because there's a step that always happens first after we get calcium present, right? So without calcium, we don't have a contraction cycle because we need calcium to be binding to reveal the actin, the myosin binding sites on the actins. So yeah, so cross bridge formation is the first step because that's what is able to occur once calcium is present. So we have myosin and actin stuck together. And the thing you have to keep in mind with this is that even though we show this as a single myosin head attached to a single actin, this is occurring along the length of the sarcomere. The entire overlapping portions of the thick and the thin filaments are going to be having cross-bridge formation. And of course, this is occurring along the entire length of the muscle cell, which is occurring, you know, across everybody in a motor unit and all that stuff, okay? So once we have cross-bridge formation, then we can actually move the filaments in relation to each other, right? So we pull the thin filaments towards the center of the sarcomere because the myosin heads have a power stroke. And the power stroke means that they actually, they rotate and they bend, but really if you just want to think about it as bending and like pointing towards the middle, the M line, right? They pull the thin filaments on either side of the sarcomere inwards and shorten the entire sarcomere. And then because one round isn't enough, we do it again. The other thing that happens during the power stroke is that they actually release the waste products of energy use. So ADP and inorganic phosphate are released, and that changes the binding of the myosin to the actin so that then we have cross-bridge detachment. So that then we can have a new ATP bind, and then that changes it so we have cross-bridge detachment. Close enough. And the nice thing about letting go is that then we can do it again, right? So then the myosin has to set up to do another round, which is where we get the fourth step of cocking the head. So we go use the energy of the ATP to get back into the high energy position. You can kind of think about this also like if you have something that's spring loaded and you like click it together so that the spring is loaded again, it's similar to that too. So that's the step that actually uses the energy, and then that lets us go back to cross-bridge formation. Then we actually make use mechanically of the energy with the power stroke, right? And then we let go with cross-bridge detachment because of the binding of the ATP, and then we do it again. So yeah, I mean, it just kind of keeps going as long as there's calcium present. Technically, we need ATP present as well, but we actually don't tend to run out of ATP. The energy usage mechanisms that we talk about later on are able to keep these cells in a good supply of ATP. So the calcium is the regulator of the continuation of the contraction cycle. Okay, so then let me go out and find the other one, and we'll talk about some smooth muscle stuff. Here. Oh, okay, right here. All right, so as far as when we're kind of comparing the different types of muscle tissue, one of the things we look at is the difference in the nervous system connection to skeletal muscle as opposed to smooth muscle. And so this is actually something that we end up covering several different times in the course. I mentioned the varicosities when we're just going over the muscle tissues. And then when we go into a little bit more detail about smooth muscle after we've learned about skeletal and contrast them, I didn't go into a ton of detail on it then because there really wasn't anything to add. but we did touch on it again a little bit when we talked about the autonomic nervous system. And that's because that's all related to each other. So the autonomic nervous system is going to be the primary nervous system control over smooth muscle. You guys know that now, we've covered it. And anything that's not specifically autonomic is going to be regulated by the enteric nervous system. And they're going to use similar synapses. So the autonomic synapses with the smooth muscle are much more diffuse than our neuromuscular junctions with skeletal muscle. So with the neuromuscular junction, we have this very specific one neuron to one muscle cell connection. And when we excite it, that's the one we excite and stimulate to contract. And with the varicosities, it's instead of the neuromuscular junction. So now we're releasing neurotransmitter from a variety of these vesicles. So whereas in the neuromuscular junction, the vesicles were releasing neurotransmitter across the synaptic cleft, here we're releasing them from these things called varicosities. So the varicosities are just little bulges of the axon terminal spread across a sheet of smooth muscle, so that we're getting this generalized contraction instead of specific contraction. And that's because when we want smooth muscle to act, it's typically acting as a unit within the wall of whatever organ it composes. And so it's just the smooth muscle equivalent of the neuromuscular junction. Was that the whole question? Yeah, so that's kind of tying together a couple of different systems that we covered, which is nice because, like I said originally, smooth muscle doesn't really get a good overview because it's not like the primary component of any one system, but we actually kind of covered two of the major systems that deal with smooth muscle after we talked about muscle, so good for muscle. That was all the questions that I was sent. So does anybody have questions? Yes, okay. All right, questions about other things. Okay, so everything that you're asking me right now is not part of the systems, right? So unless it comes up again in one of the 4 systems that we covered, it will not be on the exam. Does that make sense? I am not looking at any of the chapters before bone tissue to do this exam. But anything relevant to those things, because we definitely go back through, electrical gradients, right? I think we fleshed them out quite well in both the muscular system and the nervous system. Obviously, that's on there because it's part of those systems. Although we're not really calling them negative feedback loops necessarily, every time I talk about homeostasis, we're kind of covering negative feedback loops. And reflexes are also a form of negative feedback loop. So I'm not going to be looking back at the chapter 1 stuff for negative feedback loops, but it's being covered anyways. Okay? So if it's something that we covered again, because it's relevant to the particular system, then yes. If it's only covered in the chapters before bone tissue, it will not be on the exam. Okay? So don't look back at that stuff unless you're coming across it as you're studying one of your systems and you're like, oh crap, what was that again? I don't have details here because I took those details down before. Okay? All right. What other questions do we have? Yeah, so for cranial nerves, so you don't need to worry about locations now. We covered that in the lab exam. So you need to know now the same thing you needed to know for the nervous system exam, which is what their names are and what their general functions are. Not expecting you to know every single detail of their functions, especially the ones that do a bunch of stuff. Excuse me. But you do need to know their basic functions. Anybody else? I know there's more questions out there. All right, well, as much as I love sitting here in silence with everyone, if we don't have any questions right now, then please email them to me, preferably today, but tomorrow's fine too. The exam will be available Wednesday morning. All right. Like I said, if there is some reason why Wednesday does not work for you, please let me know. And we can make arrangements for a time that actually works for you. All right. Typically, we need to get our grades in within a week of final exams. So that's what I should be doing. Hopefully, don't see why not, but I promise that I will get them in, and until you see an actual number, I have not graded it, okay? So no need to ask me, I swear. I swear I'm working on it. Yeah, I hope that future semesters go better for everybody, because this was obviously a super weird way to try to learn stuff. So thanks for sticking with it. And yes, the entirety of Wednesday, okay? Like 7 a.m. to midnight. So it's just, if Wednesday does not work, let me know. All right. Good luck.
Thank you so much. Okay, take care.